So the main goal of this experiment is to determine the transformation efficiency of your E. coli competent cells. Okay, so how do you determine that? Mathematically, if you use the equation here, so you can determine that transformation efficiency. So what's the equation? So at the end of your transformation experiment, you will get the number of transformants. Those numbers are colony forming units. Little colonies you can count from your plates. Okay, that number will be divided by the microgram of plasmid DNA you added to the cells, okay, times volume of the transformants plated. Okay, for example, if you used about 100 picogram of POC19 DNA in your transformation, okay, so 100 picogram, if you change that to microgram, that's 100 over 1000 nanograms. So 100 picogram divided by 100 nanogram, okay, and then you have to change that to micrograms, which is 1000 times more. So actually 100 over 1 million will be the conversion of 100 picogram to microgram, right? So 100 divided by 1 million, which is 0.0001 microgram. Okay, so that's that factor there, 0.0001 microgram times how much the transformer you plated, whichever that volume is. Let's say you plated about like maybe 1200 transformants. So because there was like 200 microliter of cells plus 1 ml of urea broth medium was plated. So multiply 0.001 times 1200, and then that number will divide the total transformant you're going to get from per plate. That's how you can determine the transformation efficiency. Okay, so the transformation efficiency should be good. I'll tell you what the acceptable range of the transformation efficiency. Okay. So here are the materials that you will be using in the lab for this experiment. So you'll be given with 200 microliter aliquot frozen DH5 alpha competent cells, and they are prepared with 0.1 molar calcium chloride solution, and then you will also be given with POC19 plasmid DNA. Okay, so that's diluted like 100 nanogram per milliliter concentration. Okay, so if that's same as 100 picogram per microliter. If you use 1 microgram, for example, of that, then that'll be good for your experiment. Okay, and you will be given it the water bath that's set at 37 and 40 degrees, 2 different temperatures. If you want to test 2 different temperatures, effect on transformation efficiency, you're welcome to do that by using 2 different heat shock treatments. And then fresh LB medium that you can add when you do the recovery of the transformed cells. And then selective media at the end in the plate medium that contains ampicillin in your plate. And then sterile glass bead to spread the cells on the plate at the end of the transformation process. And you will walk inside of the biosafety hood aseptically, and you'll be spreading the cells by using glass plate beads. You'll be adding glass beads on top of the plate, and after closing the lid, you will shake them up, and glass bead will spread the cells all over the plates. And once that's done, you will incubate the plates in the 37-degree incubator. So that'll be the end of the process. So I want you to answer this question at the end of your lab. So what is transformation? How would you define that? We talked about it. And what is the main function of the cations like calcium 2+? What's the main function of that in the transformation method? What does that do? Probably it'll neutralize DNA and the membrane, right? And how do ampicillin and kanamycin kill bacterial cells? So those antibiotics that may exist in the selective medium at the end, how do they kill them? So how does ampicillin resistance or kanamycin resistant gene then may be carried by the plasmid DNA? How they help them to survive with the presence of antibiotics? Little bit of extra study you may have to do to answer the question, and then examine your result next week. So for that question number 3, so antibiotic resistance can be gained by the cells, because that plasmid DNA that's transformed into the cell may carry those antibiotic resistance. There may be ampicillin resistant gene or kanamycin resistant gene. Okay, then what do? How do they work? Okay, what are those resistant genes? What product are they produced from those genes? Okay, and how they help those cells to survive. So this diagram shows you various ways of removing antibiotics so cells can survive. Okay, so it depends on the antibiotics. Some antibiotics kills cells by binding or with maybe certain part of the cell, maybe ribosomes they target. Once they bind, and they stop the translation, and by doing so it can kill. So ampicillin is a good example of that. And then kanamycin may be attacking some other area. You can look it up and do a Google search on that. But the antibiotic resistant genes that are carried by plasmid DNA, they may produce a protein that may do these various works against the antibiotics. What is shown up here is what? Antibiotic degrading enzymes. So maybe they may be producing enzymes that can actually degrade and activate antibiotics. So then this cell can survive, right? Also, the gene may express the enzyme that can alter the structure of the antibiotics, so the antibiotics doesn't have its effect on the cell anymore, negative effect anymore, right? Another possibility is maybe this gene can make maybe affect pumping out process of the antibiotics. When antibiotics taken up, you can just diffuse it out, push it out. So by doing so, cells can survive. So there are various ways of protecting the cells. Okay, so that, I'm just telling you the possibility. So ampicillin looks like that. Okay, like I said, ampicillin can block the cell wall synthesis. Oh, cell wall synthesis. Okay, so about 90% of ampicillin resistance in the E. coli cells due to the production of TEM1, which express. So we're talking about the resistance, not the antibiotic, the ampicillin itself. Express TEM1 protein, which are commonly encountered beta-lactamase enzyme that hydrolyze ampicillin in gram-negative bacteria. So I guess resistant gene can hydrolyze ampicillin. Okay. So during transformation, E. coli cells with a plasmid DNA containing ampicillin-resistant gene, about 30 minutes of incubation is enough to select the cells with ampicillin resistance. Okay, so this TEM1 is enough made during the 30 minutes time. So then it can remove ampicillin. That's what it does. How about kanamycin, another antibiotics? It works by affecting, this is the one that can bind the ribosome. So it blocks the translation. So it binds with the ribosome and preventing the correct translation process. So if that happens, then bacterial cells cannot make their own proteins. So that's how they will die. So instead of reading the correct codon, they do not know how to read the correct codon, so they will mess up the translation process. So proteins needed cannot be synthesized if that happens. Okay, so due to that, bacterial cells can die from it. And how about the kanamycin resistant gene? You can find out how they kill them. You can do a Google search. Okay, so I hope you can find answers for those questions. That's it for the lab.